Today, we're going to have a look at the factors that affect the transpiration speed. So, this is the speed at which water is lost from the plant. So, this can be affected firstly by the loss of water of the plant, obviously. And here we have to think about evaporation, where water evaporates from the spongy mesophyll cells into the air spaces, as well as diffusion. The water gas molecules diffuse from the air spaces out of the stomata, out of the leaf. The next thing that we need to think about is the movement of water inside the plant. Now remember, water is sucked up by the roots, moves through the stem, and then goes through the leaf out of the plant. And the movement of water is mostly controlled by kinetic energy. And that's the energy of the moving particles of the water and how fast they move in different directions. Now, if we think about evaporation, evaporation can be affected by temperature. So as we increase the temperature, evaporation will increase as well. If we think about diffusion, then we need to think about humidity. So remember, diffusion always goes from a high water potential to a low water potential. So if we increase the water potential on the outside, for instance, then diffusion will occur more slowly. Then if we think about kinetic energy and the factors that affect kinetic energy, here we need to think about light as well as temperature, because these are also forms of energy and will be able to add energy to kinetic energy and increase it. So we can test the effect of these factors on transpiration through the use of a potometer. Now let's look at the different parts of the potometer first. We have a reservoir that's filled with water, as well as a beaker that's filled with water. The rest of the system is connected between this reservoir and this beaker, making the whole system full of water, a continuous tube. Water is able to move from the beaker with water all the way through the system up to our plant or our cutting. And there's no way that the water can go anywhere else. The water from the reservoir is only used to calibrate the machine. So there's a little tap that's constantly closed unless we want to calibrate it. Between the beaker of the water and our plant is a capillary tube. Now, a capillary tube is a very, very thin tube, and that makes it easier for us to measure. So at the back of the capillary tube, we can add a ruler. We will see in our capillary tube here is a tiny little bubble. This could either be a bubble, like an air bubble or an oil bubble, or we can add a piece of dye in it, like a dye bubble, to be able to measure how this little bubble moves towards our plant. We then have our plant, and our plant would be cut diagonally to make the flow of water into our plant easily. Now here we can use different plants depending on what we're trying to test. Maybe we want to test how transpiration differs in different types of species of plants. But most of the time we would like to test how different factors like sunlight and heat affects transpiration. Now, as my plant uses water and loses water through the stomata, it will pull up more water from the system. If more water is needed, more water is pulled up from our beaker, and our little bubble will then move to the left of our ruler. We can then measure this distance that the bubble has moved. We can then use this distance to measure the volume of water that was used by the plant. So we can calculate the volume by saying distance multiplied by the surface area of the capillary tube, which would be pi radius squared of the capillary tube. We're also able to measure the rate at which the plant transpirates by taking the volume divided by time. we can then compare the rate to different factors. So we can add a light source, we can put it in the dark, we can increase the temperature of the room, we can decrease the temperature of the room, we can add a fan to add wind, we can increase humidity, all of those factors, and then test the transpiration rate. Okay, so now let's have a look at how these factors actually affect transpiration. So the first one we're going to look at is heat. Now, as we increase the temperature, the kinetic energy of the particles will increase, meaning that the particles will start moving faster and bounce off each other faster. This will cause the particles to evaporate quicker from the mesophyll cells into the airspace and then diffuse quicker from the airspace to the outside of the leaf, thus evaporating quicker. So as we increase the temperature, we increase evaporation. The opposite is true for when we make it colder. So as we decrease temperature, we decrease kinetic energy and particles will start to move slower and slower, meaning that they would evaporate slower from the mesophyll cells into the air spaces and then diffuse slower out of the stomata. So by decreasing the temperature, we decrease transpiration. We can then add light, and light kind of works exactly the same as temperature. As we increase the light intensity on the plant, it increases the kinetic energy of the particles, making the particles move faster, evaporate faster from the mesophyll cells, and then diffuse faster from the air spaces out the stomata, and thus increasing the transpiration rate. So, if we increase light intensity, we increase the transpiration rate. The opposite is then true for when we decrease light intensity or make it darker. So as we decrease light intensity or make it darker, the particles will lose energy and start moving slower and slower. Thus, less of them will evaporate from the mesophyll cells and less of them will diffuse out of the stomata. So by decreasing the light intensity or increasing the darkness, we decrease the transpiration rate. To show the effect of humidity and wind, let's have a closer look at the stomata. So here we have the outside of the leaf as well as the inside of the leaf, and then our two guard cells that would make up our stomata. If there's a lot of water inside of the plant, but the humidity outside of the plant is low, it means that the water potential inside the plant is higher than the water potential outside of the plant. Water will then move into the stomata, making them more turgid and opening up to the surroundings. Thus, water will be able to diffuse out of the plant into the outside. Now, if there was a higher water potential outside the plant than inside the plant, meaning that the humidity is very high outside, then stomata will lose water to the plant as the plant would need it more. This will cause the stomata to close up, not allowing any particles of water to diffuse out, and thus transpiration will be reduced. So, in short, if we increase the humidity, we decrease transpiration. Now let's have a look at how wind will affect it. Even if there's a high humidity outside with a lot of water particles, wind will blow these water particles away, creating a low water potential outside the plant. This means that water from inside the plant will move into the stomata, making them turgid and opening up, which means water will freely diffuse out the stomata into the air outside of the plant. Let's summarize what we've learned today. So we now know that if we increase the temperature, we increase the rate of transpiration. And if we decrease the temperature, we decrease the rate of transpiration. If we increase the amount of light intensity, we increase transpiration. And if we decrease the amount of light intensity, we decrease transpiration. Now the opposite's true for humidity. So if I decrease humidity, I increase transpiration. And if I increase humidity, I decrease transpiration. Lastly, we have wind. So if I increase wind, I will increase the rate of transpiration. And if I decrease the amount of wind, I will decrease the rate of transpiration.